My dear students, welcome to Commissionerate of Collegiate Education. I am G.L.N. lecturer in charge, Department of Zoology, Government College, Anantapur. We are going to talk about genetic drift in today's class. The learning objective behind this lesson is, in this session we are going to learn about definition and also the scientist who has proposed this concept of genetic drift and at which level this genetic drift occurs, whether it is going to occur in small populations or large populations, we are going to learn. And at the end, we are going to learn about the effects and outcomes of this genetic drift. Evolution is the process of formation of complex organisms from simple organisms. There is difference of opinion among the scientists regarding the speed of evolution and forces of evolution. Charles Darwin believed that the evolution follows a snail pace because he did not know anything about mutations. And Charles Darwin believed that natural selection is the only driving force for evolution. Later, mutations were discovered by Hugo de Vries, and he opined that evolution follows a galloping pace. That means it gallops like a horse. So like that, there are many differences in the opinions regarding the time span and speed of evolution among the scientists. Later, with the passage of time, Sewall Wright proposed one concept called genetic drift. And because Sewall Wright proposed this genetic drift concept, it is also known as Sewall Wright effect. Genetic drift is defined as the change in gene frequency that is purely brought about by chance. That means it is not directional. It is totally dependent on the chance factor. That is a basic difference between natural selection and genetic drift. In case of natural selection selects only desirable genes or useful characters. Whereas genetic drift doesn't differentiate between useful character or non-useful character. It doesn't differentiate between comfort zone or discomfort zone. This always occurs in small populations. That means genetic drift is perceptible and it occurs only in small populations. Let me give one sort of comparison. Larger populations are comparable to oceans. And you know, ocean is nothing but the vast stretch of water. If you add a pinch of salt to the ocean or a drop of water to the ocean, virtually it makes no difference. So that means if the population is small, if the population is very small, then these differences, these variations, these changes that are brought about by this genetic drift will be perceptible. So like that, genetic drift occurs only in small populations. And genetic drift, even today, it is considered as one of the most important evolutionary force. The frequency of some genes may be reduced and some genes may be eliminated from the population altogether. And as I already told, genetic drift does not differentiate between favorable and unfavorable combinations. And it is an antithesis to natural selection. In genetic drift, heterozygous combinations may be changed to homozygous combinations. And sometimes the entire population may be filled up with homozygous individuals. That means there is a gradual shift from heterozygous to homozygous nature in case of genetic drift. But this doesn't mean to say that useful characters are selected, because as I already mentioned, genetic drift does not make or does not differentiate, does not make any difference between useful characters and non-useful characters. Origin of new species is also possible because of the genetic drift. And variation is the raw material for evolution. Small and isolated populations are the prerequisite for rapid evolution. And the small and isolated populations are also the prerequisites for the process of genetic drift. I will give one example. So let me review the entire concept of genetic drift along with its definition. Genetic drift is nothing but the change in the frequency of a particular gene within small population. Normally, there is a gradual shift from heterozygous condition to homozygous condition, and the entire population may be filled up with homozygous individuals. I will give one best example. When a small group of populations is isolated, such a isolated population is called founder. These founders, if they go somewhere and if they reproduce among themselves, the descendants of these founders resemble only founders but not the parental population. Parental population means the older parental population. That means they are more nearer to the founders who are isolated from the parent population initially. So here I will give the best example in the form of Red Indians. The entire Red Indian population today is totally filled up with O group, O blood group people. That means O blood group alone exists in case of Red Indians. This is called founder's principle, and this founder principle is the driving force for this genetic drift, where one character may be identified and the entire population may be filled up with this particular character in spite of its useful nature. So, like that, for example, let me take the frequency of one gene, like P or Q. The frequency may be taken to 1 or it may be reduced to 0. That means one gene may be fixed or one gene may be totally eliminated from the population. So genetic drift indicates that the gene may be either fixed or eliminated. So in such cases, one character will be totally removed from the population. So for example, in the previous example which I mentioned in the form of Red Indians, you don't find any other blood groups in Red Indians. Only O blood group is seen in case of Red Indians, because the genes for other blood groups are simply eliminated because of this genetic drift. And there is one more concept called bottleneck concept. We can learn about this in upcoming slides. Let me repeat. Fixation or loss is the result of genetic drift. Fixation of gene or loss of gene depends on the size of the population. If a small group of individuals from a population starts a new colony in an isolated region or in an isolated region, those individuals are called founders. We can compare this isolated population to today's quarantine population in COVID-19. But of course, today the people are quarantined or isolated because of the disease. But due to some factors like migration, previously a fragment of population might be separated from the parental population. If a small group of individuals from a population start a new colony in an isolated region, those individuals are called founders. Allelic frequencies of their descendants will be similar to founders rather than their ancestors. Presence of O positive group in all Red Indians is the best example. This I already mentioned. And let me visualize the concept of bottleneck. Bottleneck effect is caused by natural calamities like tsunamis, earthquakes, landslides, droughts, famines, and also floods, cyclones, like that. In these natural calamities, majority of the individuals or majority of the organisms or animals will simply die. They die a pathetic death. But there are certain luckiest survivors, and these luckiest survivors are different from the original population in their genetic makeup. Now they start reproducing. That means previously other combinations and permutations are simply eliminated, and now the survivor population will expand. The survivor population will increase. That means the genetic makeup of the newly formed individuals will be similar to that of survivors rather than the parental population. The best example for such genetic makeup is polydactylic dwarf individuals in old order Amish populations. Examples of small populations: oceanic islands, isolated communities, small breeding populations, home range or territory, and territoriality of animals. I will give one best example of this small breeding population. If you take the Scandinavian lemmings population, lemming is a rodent population that exists or that lives in mountains of the Scandinavian region, Scandinavian country. Unfortunately or unfortunately, lemmings are the only animals which have got this suicidal tendency. Normally, suicidal tendency is found only in case of human being, whereas the other organism where suicidal tendency is seen is this lemmings population. These lemmings migrate every year. They migrate to certain area and they dump themselves, or they just jump into the oceans and they die the miserable death. For this, of course, for this behavioral concept or for this behavioral concept or total process, there is wide reasons. There is a wide array of reasons. But here, the problem is when such lemmings are isolated from the rest of the lemmings, certain lemmings are killed. That means they are killed. They are being killed by suicidal tendency. But the remaining lemmings will reproduce, and these newly formed, newly born lemmings will be identical or will be similar to the luckiest survivors. So that's about the genetic drift. And here you can see the lemming population, and this bottleneck effect, which is caused by various natural calamities, is coined by one scientist by name Stebbins. So anyway, we have already made it very clear regarding lemmings. Genetic drift has no adaptive value, whereas natural selection has got adaptive value. For example, in natural selection, we have studied two moths, namely white moths and black moths. Either of them was selected depending on the conditions, whereas in case of genetic drift, there is no such directional selection. It has no adaptive value at all. Entire population may be filled up with homozygosity due to genetic drift in spite of its useful nature. Genetic drift upsets Hardy-Weinberg equilibrium. And you know that Hardy-Weinberg equilibrium deals with constancy of certain frequency of genes within a population. If Hardy-Weinberg equilibrium operates without any obstacles and without any hindrances, there will not be any scope for evolution. Whereas this genetic drift undoubtedly upsets Hardy-Weinberg equilibrium so that it acts as some sort of active force, or else it acts as a driving force for this process of evolution. So this is about the concept of genetic drift. Let me sum up. Genetic drift is nothing but the change in the frequency of genes. Normally, due to genetic drift, homozygosity will be selected, and genetic drift doesn't differentiate between useful characters and non-useful characters. The concept of genetic drift was identified or was discovered or was proposed by Sewall, wright, hence it is also known as Sewall, wright effect. And genetic drift has no adaptive value, and genetic drift always occurs in small population, and it is accelerated by founder's principles and also bottleneck effect. Thank you very much. At the end, I profusely thank CCE Andhra Pradesh for giving me this wonderful opportunity.